Hey. Hello, everyone. I would like to welcome you to today's webinar, Technique for Scalable Organoid Culture, sponsored by Formulatrix. Just want to give a quick overview of our speakers today. Today, we're going to have a presentation by Ben Mead, a postdoctoral fellow in the Scalable lab at MIT. Much of his work is doing single cell genomics and looking at cellular models of human diseases, identifying new drug targets and therapeutics. Much of his work is using adult stem cell derived organoids and applying these models to inflammation of bowel disease. Additionally, after Ben's talk, we're going to have a quick talk by Dimitri. Dimitri is a senior project manager here at Formulatrix. He's been here for over 3.5 years with a diverse background and experience. He's going to give a quick overview of the MANTIS, which was used in Ben's work as well. And after that, we'll be opening up the floor to questions. Until that time, I'd like to hand the mic over to Ben and allow him to take control.
Thanks for the introduction. I'd like to sort of start by providing a little bit of context on where I'm coming from and sort of broadly how our group approaches scientific problems. So as a postdoc in Alex Shalik's lab between MIT, the Broad Institute, and the Reagan Institute of MGH, we're very interested in sort of understanding how individual components of complex systems, like the single cells comprising our tissues and organs, work together in health and become disrupted in disease, particularly in the context of immune-mediated and infectious diseases. So to accomplish sort of these systems level studies, we apply tools including single cell RNA sequencing of tissues and in vitro systems, as well as sort of go about building bottom-up technologies for studying complex interactions through reductionist models, including organoid systems. And so in particular, we've been developing and applying different high throughput technologies to organoid systems like the one I have pictured here, with the ultimate goal of being able to provide new approaches for early drug discovery across the spectrum of diverse immune-mediated diseases. And so sort of to kind of briefly take a step back and motivate why we should all collectively be interested in building better tools for drug discovery with organoids, and therefore require techniques for the scalable culture of organoids, I kind of want to have us all consider for a moment what separates us from the world, namely our barrier tissues. If we think about barrier tissues, these are the cells of our skin, our airways, our gut, and they provide protection and interaction with the environment, including microorganisms, toxins, nutrients, while working with our body's immune system to help maintain health. And so barrier breakdown can kind of be thought along the spectrum that I have shown you here, where on one end there are things like infections and cancers, which are often accompanied by an inadequate immune response, while on sort of the other end of the spectrum, you have things like allergies and autoimmune diseases, which often arise from either defects in our barriers or immune systems and can cause overwhelming responses to minimal hazards. And so fortunately, today we really have an arsenal of therapeutic approaches to try and restore balance at this barrier tissue. And so these all sort of approach disease by either mitigating environmental exposure, things like antivirals, antibiotics, diet and lifestyle changes, or by altering the immune response. So things like vaccines, immunosuppressants, immunotherapies. However, in some instances, unfortunately, these therapeutic approaches prove totally inadequate. And surprisingly, if you sort of think about the key player in these barrier tissues, it's really the epithelial barrier itself, where there's very limited current therapeutic approaches. And so our lab in particular sees this as a potential opportunity to sort of be able to target upstream drivers of disease locally. And so if our overall goal is really to be able to quickly find new biological targets within these epithelial barrier cells and to target that biology with small molecules or other therapeutic perturbations in an efficient manner, what we really need are systems to test in that represent this biology well, and pipelines to quickly assess molecules that are enabling and provide minimally biased readouts. So fortunately, on the one end, we have things like organoid models, things including the intestinal organoids, which provide this opportunity to sort of unlock studies of the epithelial barrier. Additionally, they can be human-derived, so you can get human high-fidelity biology there. And ideally, they'll be able to provide higher fidelity representation of new unexplored biological targets. Additionally, we're able to pair organoid models with new screening pipelines We're able to achieve biological representation approaching animal models, but are sort of able to scale on the order of cell lines in conventional high throughput screening type formats. Also, we're able to use high content readouts like single cell RNA sequencing to better understand these complex and dynamic models. And finally, we can sort of compare what we see in these models with in vivo responses to sort of better understand if and how shots on therapeutic targets may miss. And so briefly to sort of get into what organoid culture is. So while organoids offer this new opportunity to study tissue biology with remarkable fidelity, the conventional culture of an organoid is often a labor-intensive, high-touch process, requiring significant time from well-trained individuals with limited throughput. And so there are multiple ways to grow primary tissue-derived organoids, and today I'll be speaking in the context of small intestinal organoids with which I have the most experience. And those can be grown with the use of either fibroblast feeder layers or on the surface of controlled biomaterial substrates, or most popularly encapsulated within biologically derived extracellular matrices like matrigel. Additionally, the media for organoid growth is often this complex combination of recombinant or cell line generated growth factors and small molecules. And so by far the most popular method for growing small intestinal organoids is with matrigel domes, which are seated onto pre-warmed tissue culture plates. They are then allowed to gel at 37 degrees Celsius, and then finally topped off with a complex media. And so I'll refer to this process throughout the rest of my talk as organoid plating, which is something we can really define by three specific requirements. And so the first is being able to have precise deposition of a cell-laden ECM onto a pre-warmed plate. And then being able to operate with small volumes of this extracellular are often quite expensive. And when working with like things like human-derived samples, you're really quite limited in the amount of biological material you have to work with in plate. And sort of the final requirement is being able to have a reasonable degree of temperature control, given that matrigel and similar substrates are viscous liquid around 4 degrees Celsius when being deposited, but require a warm surface and environment to form solidified hydrogels at a physiological 37 degrees C. And so in essence, really to miniaturize an organoid culture and thereby achieve scale compatible with conventional screening equipment, either in 96 or 384 well plate format, the main hurdle is really one of how well can you hit a target. And so creating a good matrigel dome in which we can hold organoids above the surface of a plate and enable maximal growth requires precisely and quickly depositing a cell-laden droplet onto a warm tissue culture surface, sufficiently far from any plate edge so that the droplet won't necessarily adhere and spread and lose its dome shape. And so by hand, this is a fairly trivial process, but can be time consuming. And it can be done in plates down to the scale of about 48 wells, reasonably well by hand. At 96 wells, it's still possible to achieve droplet plating by hand, but the rate of misformation and time to plate both rise significantly, and this greatly limits your overall reproducibility. Now further, if we aim for something even smaller, like a 384 well plate, it becomes pretty much impossible to achieve this by hand. And it was sort of at this point that we collectively sort of started thinking there must be a better automated solution that can quickly and reproducibly dispense droplets with high precision. And so we spent a while looking through different bench scale liquid handlers, and we're really intrigued by the possibilities that the MANTIS had offered. So specifically, the single free arm of a MANTIS would allow for precision programming of dispensing droplets across a variety of plate formats, enabling us to effectively hit that target. Additionally, because the MANTIS uses a washable disposable chip that can be fed from a pipette tip, we could work with a limited starting sample volume and not need large liquid dead volumes, thereby limiting the waste of precious patient-derived material and expensive reagents. And finally, because the MANTIS has a small footprint, we would be able to install the entire unit within a 4 degrees C mini fridge here under our bench, allowing for the plating of liquid matrigel, which needs to be maintained at around 4 degrees C. And then combine in that a preheated block to be able to deposit the chilled matrigel droplets onto a pre-warmed tissue culture surface. And so here I just sort of have a video for you of what that looks like in action. And so what you can see is our 96 well plate being plated with matrigel, effectively dispensing every other well. And so the end result of that is sort of what I have for you here in a dyed matrigel, where you can see the domes are evenly dispensed every other block in the center of the 96 well plate. And here is the undyed result, where you can see the nice formation of gels. Further, we're able to kind of scale this process to 384 well, which you can see here in a nice little video that goes. And so also here is a stitched image taken at 4x on a bright field scope, where you can see the ECM dome within the center of a well being well deposited. And so we're able to really hit millimeter precision here and get these domes to form reasonably well with a very small volume. And so not only are we able to dispense, but we can actually show that these organoids are morphologically indistinguishable from what you would get by hand. We don't see any notable differences in the morphology of these small intestinal organoids. And we also see that they're relatively well distributed within a gel. So these are just images taken from some small intestinal organoids plated in the 96 well plate format with the MANTIS and grown for about 6 days. You can see they're relatively well distributed, and individual organoids have that expected cryptovillus morphology going on. And so to kind of get a little bit more quantitative of what this plating technique has really enabled us to do, you can see that we're actually able to substantially reduce the reagent requirements we have, including the matrigel volume required per well, the total media volume required per well, and the organoid seeding number required per well. And then further, using the MANTIS, we've actually been able to significantly reduce the time to plate each well as well. And so what this ends up looking like, if we compare directly what we see in a 96 well plate plated with the MANTIS relative to what we can achieve in a 48 well plate by hand, using the MANTIS is about 6 times faster in plating each well. It enables you to get about 4 times more wells with the same starting organoid number that you would seed, and you're able to do about 2.5 to 3 times less reagent per sample. If we step this up one level to what we're able to see with the 384 well relative to the 48 well plated by hand, you're able to get 8 times faster plating per well, an order of magnitude more wells with constant starting material, and also an order of magnitude decrease in reagent requirements per sample. And so in total, what this has really enabled us to start thinking about is how our miniaturization approach lets us start doing things with multi-perturbations, starting with limited biological material along the lines of something that you'd be able to achieve with like a single patient biopsy or organoids derived from a single patient biopsy. So really getting into the idea of personalized medicine. And so to kind of bring it all together, what I was going to share with you is an example of how we've been able to use this miniaturization approach to actually accomplish pilot small molecule screening activities using small intestinal organoids. And so here, basically what we did was we performed a pilot small molecule differentiation screen, where over a 6-day period, we did lineage-directed differentiation and sought to identify tool compounds that might actually be able to enhance differentiation of specific secretory cell existing within a small intestinal organoid called the PANIS cell. And PANIS cells are the primary antimicrobial producing cells of our small intestines, and they're critically important for barrier function. So along the lines of the research we like to think about in the lab. And so how we were actually able to do this was we built an assay consisting of first expanding murine organoids enriched as intestinal stem cells for 6 days, at which point we then replated these into 384 well plates, and then pinned in a library of small molecules with an additional media change and pinning at 3 days, and a final assay at 6 days post-plating. And so to measure a proxy of PANIS cell differentiation, we adapted an assay to measure secreted lysozyme, which is a canonical PANIS cell produced antimicrobial protein. And we measured this both basally and following secretory stimulation with an agent called carbamylcholine, or I've abbreviated here CCH. And then finally, we paired this functional measure with a measurement of total well ATP as a proxy for cell number to sort of normalize effects that we might be able to measure in here. And so in total, this proof of concept screen used a library of about 450 small molecules across 4 doses and with 3 biological replicates, resulting in over 5,000 total wells. Something that's sort of a scale that would be otherwise impossible without having miniaturization and automation, and totally inaccessible to the sort of conventional method of intestinal organoid plating. And so in this primary screen, we used a robust statistical measure known as the strictly standardized mean difference, abbreviated here as SSMD, to be able to classify our hits. And we considered increases in both the basal and secreted lysozyme, which you can see are well correlated in our primary data set here, and are suggestive of PANIS cell enrichment. And so those hits were the ones that we highlighted in yellow. And when we look within those hits, we can further restrict our consideration to the hits with the largest measured effect, many of which were significant only in the functional assay and not both in functional assay and cell number. And to us, this suggested that there's specific differentiation effects going on. And so while I won't really be able to dive into the details of the follow-ups that we've gotten from this primary screen, we're really excited to have started doing some follow-ups on a number of these compounds that have suggested that we're actually able to sort of start delineating new biological targets for consideration in panacell-directed differentiation, all enabled by sort of this miniaturization approach that was previously inaccessible to us. And so with that, I'd kind of just like to wrap up by sort of reviewing the key elements of what has enabled us to do this miniaturization. So first of all, it was sort of being able to define what the key elements were, that of having precision plating, low volume usage, and a relatively good degree of temperature control. And using that combined with the MANTIS, we were able to sort of build a bench scale solution to miniaturize an organoid culture, and with that achieve scale amenable to high throughput screening activities. And so going forward, in turn, we really think this will empower more functional studies of barrier tissue hypotheses across health and disease. And we're really poised to sort of start building out these technologies and grow our efforts. So importantly, we know that organoids are a powerful tool, but we also don't yet, as a field, fully appreciate their limitations, suggesting that there's probably a lot of meaningful work to do in kind of better understanding where these models are best applied and building out better tools for discovery. And so along these lines, we're actively engaged in improving our technologies to enable greater scale, more modes of measurement, and improved model fidelity. And we're actually really eager to engage with the broader scientific community and those of you who are listening in today to think about areas where these approaches could be most significant in the identification of new therapeutic opportunities. And so with that, I'd just like to briefly close by sort of acknowledging the team that I've had the huge privilege of working with every day, including Alex and the whole lab pictured here, as well as the generous funders of all of the work going on in lab, and in particular, GlaxoSmithKline, who has so generously supported my postdoc fellowship. And so with that, I'd be happy to hand it back to the team at Formulatrix and look forward to answering some of your questions. Thanks.
Great, thanks a lot, Ben. So we're going to have Dimitri give a quick overview on the MANTIS, which was used during your work, and then we'll open the floor to any questions. So let's see if we can get Dimitri on the line.
Hello. I'll give a quick overview of the MANTIS instrument. MANTIS is a single-channel, non-contact microfluidic dispenser. That means that it can deliver a single reagent into a single well at a time. The liquid path is entirely confined to the disposable so-called chips, which are in fact the microfluidic diaphragm pumps. So that means that there is no crosstalk and cross-contamination between reagents and no necessary washing. It is capable of delivering volume starting with 0.1 microliters and up. And at that volume, it is incredibly precise, industry-leading, less than 3% CV. The number of chips that MANTIS can support, depending on the configuration, is between 6 and 48, which makes it quite flexible for multiple workflows. As far as solutions are concerned, it can handle aqueous solutions up to 25 centipoise, which translates to approximately 60% glycerol at room temperature. So it's pretty well suited for dispensing master mixes and other viscous things. We also have a line of chips that are specifically delivered for handling organic solutions. All of that comes in a tiny footprint of approximately 1 cubic foot. And MANTIS is very flexible. It can accommodate a wide range of inputs, starting from pipette tips and ending with half-liter bottles. It can deal with almost any labware as long as it fits in the SBS footprint and has a fixed pitch. And it integrates with other instruments, including larger liquid handlers, where it can complement the base technology with non-contact bulk dispensing.
Great, thanks, Dimitri. All right, I'd like to open up the floor to any questions. If you guys have any questions, by all means, please write them into the list. Let me see if I can pull out the list here to see which questions have come through. Looks like we have a couple. So first on the list, looks like we have, so this one would be for Ben. When you use this technique to plate in a 384 well format, it says, how long does it take to make a complete plate and how accurate is it? And is every well plated correctly?
Great question. So in terms of plating a full 384 well plate, in my hands, it takes about 5 minutes, which isn't the fastest thing in the world, but I think what you really have to appreciate is sort of the scale you're able to achieve. And on a per well basis, it's pretty quick. Practically speaking, 5 minutes per well is enough for me, fast enough for me to get some work done. But I think if you think of a screening campaign on the order of like hundreds of thousands of wells, it's quite time intensive. And then I think the second part was about accuracy or wells plated correctly. And so it's really a matter of how well you actually go in and program in the plate using the software. And so what we've been able to do with a reasonable degree of fidelity is get about like 95% of the wells plated correctly. And so in that aspect, I think it's quite good, especially considering that it's all but impossible to achieve that by hand. And that's with a standard 384 well plate with a 1 microliter gel.
So the question comes in, and this is again for Ben. What did you use to enrich your organoids with stem cells during organoid expansion before enriching them with Paneth cells?
Yeah, so what we used in that case was a method we'd actually published previous to me joining my old graduate lab in Nature Methods in 2014, where effectively, and this was in mouse-derived small intestine, you're able to add 2 small molecules targeting Wnt and Notch pathways, CHIR 99021 and valproic acid. And so by adding those, we'd previously shown that you can actually get a fairly substantial enrichment in the stem cell compartment. And so we really use that to basically be able to massively expand the organoid population prior to any screening campaign and sort of provide like a blank surface to do a differentiation screening.
Okay. So questions come in. Could you discuss performing single cell RNA sequence on organoid model systems?
Yeah, so that's a little bit out of the context of today, but I can briefly touch on that, and maybe it's something that I think you could reach out to me under an email, and I'd be happy to discuss a little bit further on what we've started to think about. But we've done a couple of things. So we use a platform that our lab has developed called Sequel, which is relatively inexpensive and in-house platform for doing single cell RNA sequencing. And in that context, we've really started to sort of get at questions of what's the true heterogeneity of different organoids, and specifically how do some of these different chemical perturbations you do to an organoid influence its overall heterogeneity, and what different cell types in there actually represent. And so I think it's sort of an open question of when you look at specific cells within an organoid, while when you look at like a few immunofluorescence markers, it looks like you have good representation of different cell types. We've seen that it really requires going in and doing a more holistic profile to sort of gauge how mature those cell types are and what are their functional capacities. And so those are the kinds of questions we think about with single cell analysis.
Great, thanks. So are the organoids built in the matrigel, or are they made separately and then added to the gel before plating?
Yeah, so organoids are typically derived from primary tissue. So what you really do is there's a bunch of different isolation protocols out there. What we've done conventionally is sort of adapted a protocol that was originally published, I think, in 2009 by Sato et al., where you can do crypt isolation from primary intestinal tissue samples. And then what you actually end up plating are those intestinal crypts, which then, when given the matrigel, they're suspended in the matrigel. As well as the sort of complex media, they actually start to sort of self-form and grow these complex structures.
Great. Another one for Ben, but this is with regards to the MANTIS. How long did it take to optimize the MANTIS for organoid seeding, the whole process?
Yeah, so I think once we knew what we wanted to do, it was relatively straightforward. Fortunately, the software for working with the MANTIS is quite user-friendly. The most frustrating part, I would have to say, is just tuning it to get precision within individual plates. So every time you change a plate from a different manufacturer or slightly move something around, you sort of have to reposition the arm. But I would say, after having done it a few times, I can program a plate in within half an hour and get good results with that.
So I believe you covered this, but How do you ensure even distribution of the organoid fragments during plating across the different droplets when using the MANTIS?
Yeah, so that's an excellent question. And I think it's something that if anyone wanted to adapt this technique, they would sort of have to empirically determine. And so one of the things you sort of contend with is while you're doing this dispense through a pipette tip, the matrigel is still a viscous liquid, and so there's some settling effect in there. What we found works best is sort of if you dilute the matrigel optimally with the basal media you're working with, you can actually get a consistency where you're able to sort of plate without a bunch of redistribution during the plating. And so what we're able to see is reasonable variability. And so one of the important things I think when you're doing this is always to have something to sort of normalize for cell number across wells. Intrinsic to any screening campaign you do, you will see well-to-well variability. And so there's a couple of ways to sort of optimize that.
What is the rationale for seeding organoids in a sphere?
Yeah, so I think there's a couple of different ways that have actually been shown previously to sort of achieving organoid culture at scale, not necessarily all requiring a sphere. I think if you think about a sphere though, it's a quite optimal shape in terms of surface area, right? So when you put a cell in a culture well, you really want to be able to ensure that you have maximal exchange of that cell with nutrients and reagents that are housed in the media. And so by putting that in a sphere, you're not only able to sort of keep the organoid cell structures lifted off of the plate surface, because if the organoid actually touches the plate surface, that can be detrimental to its phenotype. So by plating in a sphere, you not only get it to be suspended off the surface, but you also sort of ensure that there is sort of optimal exchange of nutrients with the media. And you sort of minimize the total volume of reagent that you need to use, right? An alternative would be just filling the bottom of a well with matrigel and putting organoids on top, and that would work similarly, but you sort of burn through a lot more reagent in that process.
This one I think is a good one for the system that you're using. What kind of variability do you see between wells in terms of number of organoids, etc.?
Yeah, so I think I touched on that a little bit already, but it's very context dependent, I would say. So any system you're working with, it's quite context dependent. So for us, we've been able to optimize sort of the ideal number of organoids to plate per well. And with that, I would say the variability is on the range of 20 to 30% per well, which sounds substantial, but with a good normalization strategy, it really isn't a big enough hurdle to sort of be able to pull hits out of.
Okay, great. So with any other questions that we're not able to get to, or if you have questions after this presentation, feel free to send them to myself. I'll try to get those on to Ben or Dimitri respectively to get some answers for you guys. And we will be sending you a link to this presentation after we're finished here. So I want to thank your time again. Thank Ben for his time and Dimitri. And with that, we'll finish today's webinar, and you guys have a good day. Thanks. Bye.